My name is Cesar de la Fuente. I'm a presidential associate professor at the University of Pennsylvania in the United States. Yes, in my lab, we use the power of machines to accelerate discoveries in biology and in medicine. Specifically, we work on antibiotic discovery. So we try to use AI and computers to accelerate our ability to come up with new antibiotics. The reason that this is really important is that antimicrobial resistant infections are increasingly killing more and more people every single year. They're currently associated with about 5 million deaths per year in the world. And I think this is probably the greatest existential threat to humanity. And so with AI, we can really accelerate our discoveries in this area. And we've shown that over the last decade or so. And so really that's what we focus on in my lab. Receiving the Fleming Prize is a tremendous honor. It's really a testament to a lot of the work that people have done in my lab as well, and my mentors and colleagues and my family that have supported me over the years. Particularly special to be receiving this award after one of my scientific heroes, Alexander Fleming, who of course discovered penicillin in 1928. We've tried to do what we can, and now instead of finding antibiotics in nature like he did in a fungus, we find them on the computer by using AI. But we hope to contribute something minimally as important as what he did, and it's really a huge honor. So in my lab, we basically try to teach computers and machines to come up with entirely new types of antibiotics to treat infections that are currently untreatable today in the world. And in addition to that, we use algorithms to try to accelerate our ability to discover new antibiotics. And so the way we do that is we have these AI systems that can systematically mine all of biology. And by doing that, they can find sort of hidden molecules that otherwise the human brain would not be able to perceive or see. And in doing so, we've been able to dramatically accelerate discovery. So one of our latest AI systems is called Apex, and it's trained on our in-house data set, which contains thousands of molecules with the respective antimicrobial score. So we actually do the experiments where we test them against bacteria, we see how well they're able to kill them, and then we record all of that data, and the model is trained on all of those data. And so it's really good at them picking up or identifying sequences within genomes or proteomes that might look like potential antibiotics. And then you can do that really at digital speed. So you can do that in a few hours, in minutes, really. We have people sending us emails all the time, asking us to mine their genomes or the metagenomes of interest, and sometimes within minutes we can return the results back to them. Which is always really fun to do. But it's really the power of AI. We can do things dramatically faster than we could many years, just 5 years ago. Yeah, I think sharing our findings is crucial, really, to encourage innovation and to encourage the scientific community to advance the field of antibiotic discovery. So recently, for example, along with our colleagues, Luis Pedro Coello and his team, we released a whole data set of almost 1 million new sequences, new antibiotic sequences, onto our website, onto a data set that anybody can access to. And our ultimate goal is we're only one lab or two labs working together. So we think that if we put it out into the public and into the scientific community, that'll be thousands or even more scientists trying to advance those molecules further. So I think it's just a way of accelerating discovery and hopefully benefiting the scientific community and benefiting humanity ultimately. It's going to be very hard for one person or one lab to solve the antimicrobial resistance or AMR problem. And so I think we have to be humble about it. And I think by just simply sharing everything with the world, we'll have a better chance at really tackling and addressing AMR. It's an incredibly difficult problem. Even Alexander Fleming, in his Nobel Prize address, he mentioned, that bacteria could easily become resistant to penicillin, and we're seeing that with all the other antibiotics that we have at our disposal. And so I think it's really important to work as a community. And I think microbiologists have historically been really good at that. So let's continue doing that. Let's continue working together, collaborating, sharing resources, and again, and welcoming people from other fields too. I think there have been several highlights for sure. I think as scientists, we're always seeking this moment where we discover something that nobody else knows in the world. And those moments happen very rarely, of course, but we're always kind of seeking those intimate moments of discovery. And I've had a couple in my career. One of them probably was when we were able to successfully design an antibiotic on the computer, exclusively computationally, that then was, once we synthesized it, it was capable of clearing infections in test tubes, bacterial infections, and also in mouse models. So that sort of opened our eyes into, okay, this can be an entirely new field where we can use AI for antibiotic discovery. Another one maybe was when we, by using machine learning, we were able to identify therapeutic antibiotics in ancient biology and extinct organisms. For example, in ancient humans like Neanderthals in their genomes and proteomes, or by looking at all of ancient biology, including through the Holocene and the Pleistocene, we're able to identify compounds in things like the ancient elephant and the woolly mammoth and giant sloths and creatures from the past. So I think that was also another extremely exciting moment where here we were looking for the very first time into sort of ancient biology, right, as a source of new bioactive compounds, and so that was maybe another one, and then maybe like some of the work that we've done, just generally mining the tree of life to try to find new antibiotics. I mean, I think that has also been a huge team effort that has been really, really amazing to just discover millions of new compounds that we think play a role in the immune system, by the way. So I think it's really uncovering a new branch, perhaps, of the immune system that is peptide-based, sort of like small protein-based. So I think that has also been really, really rewarding and really, really amazing to see. I'm really fortunate to be able to work with an amazing team. We really have a transdisciplinary environment where we transcend individual disciplines. And just to give you a sense of this, right now in my lab, we have people that come with a computer science background, people that come that are chemists, people that are physicists, people that are biologists, microbiologists, of course, engineers. So it's really many, many people coming from all walks of life into my lab. And by the way, they come from all different places of the world as well, but also with different backgrounds, different life stories, different ways of thinking. And they come into my lab to work together as a team. And they all bring different perspectives that add to different ways of thinking and coming up with new ideas that all contribute to the overarching goal of the lab, which is accelerating discoveries by using AI, in this case to try to come up with new antibiotics. But I mean, really, I'm incredibly fortunate to be able to work with people that know a lot more than me about many things. So I actually get to learn every single day from them, from their own backgrounds, their own ways of thinking. And that ultimately just leads to breakthroughs and advances science and advances innovation. And it's really I'm extremely sort of lucky to really be able to direct that orchestra that we have in my lab, and to welcome people from the best people, the best talent from around the world to come to the United States, to my lab, to work on these really, really big problems. Well, thank you, Gordon, for the very kind introduction, and really I couldn't be more honored to receive the Fleming Prize. Let's see here. Alexander Fleming is really one of my scientific heroes growing up, and so it really was a tremendous honor when I was notified that I would be awarded this prize. So thank you again to the organizers and the committee, the prize committee, for selecting me. I'd like to reflect on this quote from Fleming himself when he was awarded the Nobel Prize. He basically had the foresight to predict or to already notice that antibiotic resistance happened rather quickly, right? Of course, he found penicillin, the very first antibiotic, in 1928 in nature, produced by fungi. So today I'm going to tell you a little bit about how we can find antibiotics, maybe instead of in nature, on the computer. Of course, I don't need to really introduce this. I believe that antimicrobial resistance is the greatest existential threat to humanity. These are bacterial infections that are, many of them, untreatable. They're currently associated with about 5 million deaths per year in the world, and the projection is that by 2050, that number is going to go all the way up to 10 million people per year, surpassing every other major cause of death in our society, as we can see in this graph, including even cancer. So it's really a huge problem where there's a huge gap in innovation and in, like Gordon was saying, in trying to come up with new classes of antibiotics or new types of, new ways of looking at the problem. So that 10 million deaths per year, they corresponds to about 1 death every 3 seconds. The other thing with antibiotic discovery is that it's really slow. So it can take up to 6 years to come up with preclinical candidates. It can take over a decade to really develop a drug, and it's also really expensive. And so we thought a number of years ago, we thought that instead of using the traditional paradigm, why not try to use AI to accelerate this process of discovering new antibiotics. Like I mentioned, the traditional paradigm in this field has always been to go around nature and try to find new compounds in organic matter, things like soil samples and water samples. As you can imagine, this is a really painstaking process that relies on trial and error and oftentimes does not yield any good sort of candidates at the end of the day. So instead of doing that, we had this idea of taking advantage of the decades worth of biological data that we have at our disposal in the form of genomes, metagenomes, and proteomes, and to conceptualize all of biology, all of that data as code, right? Nucleotide code, amino acid code. And if we conceptualize all of biology as code, then we can try to develop algorithms to try to sort through all this tremendously vast code to try to find new potentially bioactive molecules. So it's sort of like going with a magnifying glass and trying to zoom into biological data to try to find new potentially useful molecules. And sort of how we've done it is we can scan individual proteins, like I show here in this diagram, to try to find those ones that have fragments that might be antibiotic. We had to come up with a paradigm for this. And so first, and a lot of what I'll explain today, it underpins this idea of developing our own data sets where we have an objective function, which is antibiotic activity. We want to find molecules that have antibiotic activity. And in order to measure success, we need a reliable assay, experimental assay, right? To benchmark against. And of course, in microbiology, we have this great assay called the MIC assay that measures the antimicrobial activity of any given agent when we expose it to bacteria on a test tube or a 96-well plate. So we're really lucky to have this, right? And then once we've built these data sets internally in my lab using experiments, then we can use them to train AI models. To then be able to identify, discover, and generate new compounds. So this is sort of the idea that we had a number of years ago. So now I'm going to tell you a bit chronologically the progress that we've been able to make. So in order to identify antibiotics in biological data, first we took inspiration from image and speech recognition algorithms. But of course, instead of recognizing sounds or faces, we wanted to recognize molecular patterns that were associated with things that might be potentially antibiotic. So it's quite different. So we started by mining individual proteins to see if we could find fragments encoded within the protein that might be antimicrobial fragments. So a protein, we can show it in 3 dimensions, but we can also dispose it in 2 dimensions with the amino acid code here. And the algorithms, essentially, they run through the code, and they identify regions that are predicted to be antimicrobial, and these are given by these different colors here. Of course, this is only a prediction. It's a theoretical work, but it's powerful because it allows us to then explore individual proteins and identify within that complexity regions that might be antimicrobial, for example, this region here highlighted in yellow. And then in the lab, we can use synthetic biology tools and chemistry tools, and we can extract that fragment from the protein to try to develop it into a potentially useful thing, right? So we did this for a number of projects. We were mining individual proteins to try to find encoded antibiotics. But then with advances in compute power, we realized that we could scale up this whole process. And so we embarked on this journey to try to mine the human proteome for the first time as a source of antibiotics. The human proteome is all the proteins encoded in our genome, so it's essentially our entire genetic code. And that's what we did. We developed a really simple algorithm to do this. Some of the details are there, but it imposed a huge scaling problem, right? Because we were used to just mining one protein at a time, and here we ended up mining 42,000 proteins because we took into account isoforms as well. So it was a huge scaling problem that we had to face, and we weren't sure how long this would take. And it was amazing to see that our very inexpensive, computationally speaking, algorithm was able to essentially browse through our whole genetic code, through all the genetic information in our body in just 1 hour. So this was really amazing to see, and it really convinced us that this could be really a powerful tool, right, of digitally mining genomes and proteomes really, really rapidly to try to identify bioactive compounds. So this essentially what it meant is that it enabled us to identify thousands of new compounds that we call encrypted peptides that are hidden or encrypted within larger proteins. And then out of those thousands, we synthesized 56 using chemistry in the lab. And we tested them against bacteria. We got basically around 62% hit rate, meaning 62% of the encrypted peptides that we synthesized were actually active against either gram-negative or gram-positive bacterial pathogens. So some of the things that we found, like I mentioned, is that a lot of these peptides had broad-spectrum activity, right? So now, at this point, we hypothesize that maybe they play a role in host immunity. Maybe they're previously unexplored or unknown sort of bioactive peptides that were always there, but they were not previously recognized to play a role in the immune system. And we're continuing to test this hypothesis. Many of them are predicted to be cleaved off by proteases from their parent protein, which oftentimes has a different function. So it doesn't play a role in the immune system or in innate immunity or anything like that. And many of them, actually, the vast majority of them in this human proteome work of the peptides came from non-immune proteins. So this led us to actually come up with this hypothesis that we call the crosstalk hypothesis, where we pose that a lot of these peptides, since they come from non-immune proteins, we pose that there are a lot of body systems beyond the immune system that might be contributing either directly or indirectly to host immunity. And that's this sort of crosstalk between, and just to give you an example, we found peptides, for example, in the nervous system, the digestive system, the cardiovascular system, and so on. And so we think that upon infection, a lot of these peptides get, many of them cleaved off from parent proteins that have a completely different role, and then they contribute to host immunity, and many, and perhaps other physiological processes as well. And we just published a paper in Trends Biotech, kind of testing this hypothesis, and we found that many of them have these sort of antimicrobial roles and also immunomodulatory properties. And then we tested them in preclinical mouse models of infection, some of the lead compounds, and we saw that they were capable of reducing infections caused by Pseudomonas aeruginosa and Ebomani and other pathogens. In this case, this is an abscess infection model. And we also saw synergy. So actually, if we combine different encrypted peptides that are produced in the same area of the body. They synergize with each other to target pathogens. So we think that that might be the way that they operate in vivo, in an in vivo system, again, as this sort of first line of defense against pathogens. We also found that some of them are expressed physiologically. These are 4 examples. And they're expressed at different levels, from low to medium to high, and in different parts of the body, like I mentioned, cardiovascular system, digestive system, nervous system, and so on. And it's really interesting that some people have followed up on this work in addition to the work that we've done in my lab. And just a month ago, we had a preprint from my lab, and also there was a group from Israel publishing in Nature that the proteasome, which is the degradation machine of the body, also a lot of the proteins that it degrades, they generate bioactive encrypted peptides that play a role in host immunity, and that they're actually activated upon infection. And so sort of this hypothesis seems to be supported by data from other groups as well, so it's really exciting to see that. So this work, it also sparked a lot of new ideas in my lab, one of which was the fact that we're finding all these thousands of compounds in the human proteome. It probably means that they're not just there. And so we came up with this idea that maybe we would be able to find similar peptides all throughout evolution and across the tree of life. And since we had done this work with the human proteome, we decided to look at our closest ancestors to see if we could find also bioactive peptides there. Our closest ancestors are Neanderthals and Denisovans. And in the process of thinking about all of this work, we came up with a framework for identifying new molecules that we call molecular de-extinction, whose ultimate goal really is to identify molecules all throughout evolution, to then be able to synthesize them in the lab, to be able to learn how certain mutations that occur over time or levels of conservation influence their function. And their function can be antibiotic properties, which is what we've been focusing on, but it can also be immunomodulatory properties, ability to dampen inflammation, it can be anti-cancer properties, you name it. So this is really a framework for coming up with new molecules and to be able to study them throughout the course of evolution. So going back to ancient humans, to be able to discover or identify bioactive compounds in archaic hominins, we developed this machine learning pipeline that we call PanCleave, that at a very high level, what it does is it takes all the proteins from Neanderthals and Denisovans, and it chews them up in little fragments. And then we have 2 filters, one of which is a machine learning filter, the other one which is a human expert filter, to predict whether those fragments will be antimicrobial or not. Sort of that's kind of how we see here in this pipeline. And I think it's really important to highlight that every critical decision that we make in my lab, we take into account the recommendation made by the machine, but also the recommendation made by the human scientists, the chemists, the biochemists, the microbiologists that are working in the lab. And so I think this sort of that critical synergy between machine intelligence and human ingenuity is going to continue. So this ML platform, it enabled us to identify therapeutic molecules in ancient humans. Here we also explored modern humans as a control. Here are some of the encrypted peptides that we found in Denisovan proteins and Neanderthal proteins. Of course, this is only theoretical work. So then we needed to synthesize a lot of these compounds in order to validate them in the lab to see if they were able to actually kill bacteria. And some of them did in vitro, and some of them also did in vivo in two different preclinical mouse models. The one on the top is the abscess model. The one on the bottom is a deep thigh infection model. And the top hit was Neanderthalin. We also had to come up with new nomenclatures for all these compounds. So the convention that we are following is that we name them based on the organism that produces them. So Neanderthalin comes from Neanderthals, right? And that was the top compound, but none of them were highly bactericidal. They were mostly bacteriostatic. So they were not, the next antibiotic, right? None of them. But I think importantly, this convinced us that applying this framework of molecular de-extinction was possible, and that we were able to demonstrate that you could do it in a practical fashion, right? You could apply it and then actually identify promising molecules. So then this gave us the courage to ask maybe a more ambitious question, which was, why not just sample all of ancient biology to see if we could find bioactive antibiotics there? But in order to tackle this, again, this was another scaling problem. Here, we're exploring 3 organisms, right? Neanderthals, Denisovans, and modern humans. But if we want to sample all of ancient biology, that's all of known ancient biology, that's hundreds of organisms. So we needed to develop a more powerful AI model. So this was work that took several years, but we developed Apex. This is the architecture of Apex. And the really powerful thing about Apex is that you give it an amino acid sequence, and it predicts the antibiotic function directly from the sequence. So it's a sequence to function prediction model. It doesn't take into account structure at all. So it literally, you throw at it an amino acid sequence, and it has to predict what that sequence will do out in the world. In this case, kill bacteria, right? It's a hard problem. And that's kind of the goal. That was the goal of training and developing Apex. A bit of a background story with Apex is that initially, when we started this work, we knew we needed to develop our own in-house data set experimentally to be able to train Apex. And I had initially calculated that it would take us about 2.5 years to sort of generate enough data to train the model reliably. So I'll tell you a bit of the background story of the failures, because this is also important, I think, in science. After 1 year of collecting data, we tried to train Apex. It didn't work. The performance metrics were pretty bad. After 1.5 years, it also didn't work. We tried again. Then at 2 years, we tried again, and it failed again. At 2.5 years, which had been my prediction, we tried again, and it failed again. So then I started worrying that perhaps my calculations had been off by maybe an order of magnitude or something, in which case it would have been unfeasible to do this project in the context of an academic setting and PhDs and so on, right? So it actually, it took some convincing to my team to continue building this data set. And after 3.5 years, we had generated enough data to actually train Apex successfully. But there was a lot of hesitation and uncertainty all along the way. And I just thought I would mention this because in science and in research, oftentimes we don't talk about the background stories. Okay, so Apex, it enabled us to sample all of ancient biology. Here are some examples and the molecules that we found. And in this diagram, you can see how we were able to explore digitally all organisms from the Holocene and the Pleistocene. And here I'm showing organisms, representative organisms, where we found some of the top candidates. It includes penguins that were extinct relatively recently in the 50s. It includes magnolia trees that disappeared throughout evolution, ancient elephants, woolly mammoths, giant sloths, and many other sort of creatures that disappeared throughout evolution. So digitally, we can now sample all of this ancient genetic information to try to find potentially useful solutions to AMR. To give you a sense of the scale of this project, these are all the organisms that we sampled. Where you see active encrypted peptides or EPs, those were actually synthesized and validated in the lab. So generating this figure was really, it took a lot of effort, even though it's just one graph. And then we validated them in preclinical mouse models of infection. And here are, again, the skin abscess infection model on the top, and here the deep thigh infection model. And the top candidates basically came from the woolly mammoth, the ancient sea cow, the giant sloth, and the extinct giant elk. And the really exciting thing here is that we did see bactericidal activity in these 2 different mouse models that was comparable to polymyxin B, which is a last resort antibiotic. So it was really exciting to find some candidates in this case that were highly effective in these difficult mouse models. So this was highlighted by the editors of the journal, sort of this concept of exploring ancient biology to try to find new antibiotics to tackle AMR. So you might be wondering why molecular de-extinction. So I'll just tell you briefly a couple of things that we've learned so far. So we've been able to tap into new sequence space. So if you do sequence space visualization of the molecules that we found in ancient biology, And when you try to overlap it with living biology, a lot of it, some of it overlaps, but not all of it. So we've been able to explore new kinds of sequences. I think it expands the way we perceive molecular diversity and what is possible in terms of bioactive peptides and bioactive compounds in biology. And like I mentioned, we think that these peptides that we're finding, they play a role in immunity throughout evolution, so we're continuing to explore that. And finally, I think it's interesting to think about the potential of bringing back molecules from the past to address present-day problems. As you can imagine, a lot of contemporary pathogens that exist today, that kill people today, most likely have never seen some of these compounds that we're identifying that existed or were produced hundreds of thousands of years ago. So it's a good way of sort of identifying a new treasure trove to try to tackle problems that we have today. So a couple of ramifications from this work. One, from a bioethical standpoint, this is something that occurred to me when we were initially uncovering the compounds in ancient humans, which scientifically was exciting, but then I started worrying whether it was okay for us to synthesize some of these things using chemistry. And the reason for that is that some of the compounds, not all of them, but some of them, when we do sequence homology, we don't find them in living biology, which means that as far as we know today, some of them basically were extinct throughout evolution and are no longer expressed or produced today in the world around us. So is it okay for us to synthesize them in the lab, right? So we've been consulting with bioethicists to make sure that we innovate, but that we do so responsibly. So one of the things that we've integrated into our workflows is that if we identify sequences that might look like a biotoxin or something, we just don't pursue that kind of work. So that's one sort of conclusion from the conversations with bioethicists. The other thing is on the patenting side. When we were, I went to the patent office at the university and I asked them whether some of these compounds might be patentable. And I'm aware that natural compounds are not patentable, right? There was a legal case where it was decided that natural sequences belong to humanity, so you can't patent them. But then I asked the patent lawyers, but what about these ancient sequences, some of which no longer exist, right? They were naturally produced a long time ago, but they're not present in the world today. So this is creating a bit of confusion amongst patent lawyers, and it's quite funny to see them fight with each other about whether we should patent these molecules. But yeah, it's opening up a whole new area of patent law, essentially. We've published this opinion piece in case you want to check it out, on sort of musing about some of these concepts, some of these things. In terms of next steps, so basically we want to mine all of the world's biological information. We think of biology as a source of information that can be sampled with algorithms. And we want to basically mine the whole tree of life. And initially we started with eukaryotes in the form of the modern and ancient humans. So then we wanted to see whether we could explore the microbes of the world to see if we can find antibiotics there. So I'm going to tell you briefly about two collaborations where we did this. This is the first one with our colleague Luis Pedro Coello and his team, where we mined the global microbiome using machine learning. The global microbiome is essentially not all, but a great representation of the microbes that exist on our planet that come from all sorts of places around the world, including corals, ocean, soil, teeth, pets like cats and dogs, really everywhere. I'm talking about almost over 60,000 metagenomes and almost 90,000 genomes. And using machine learning, we're able to identify almost 1 million new antibiotic compounds by looking at all of this microbial dark matter. And remember, it's really hard to find one antibiotic. And here on the computer, we're identifying millions of them. We've released all of this publicly, so it's open access through this website that we call AMP Sphere. So everybody everywhere has access to all these compounds. And we did that in the hopes of really accelerating progress in the antibiotic field. We are only like 1 or 2 labs, right? And we believe that if many, many more labs start trying to characterize and move these compounds further, we may be able to make a lot more progress. And by the way, we synthesized 100 of them. 79 of them were active in vitro, and then in vivo, in preclinical mouse models, we also saw some hits. Again, we had to come up with new names for all these compounds that we're finding in different microbes. So this is one collaboration, and the second one is with our colleagues at Amibat's lab at Stanford, where we mined human microbiomes as a source of antibiotics. So we mined almost 2,000 microbiome samples. And we thought that microbiomes were a great place to look for new antibiotics because these are microbes that have to coexist amongst millions of other microbes, and they have to take over a particular ecological niche and space. So in order to do that, they have to sort of outcompete each other. So we thought this would be a great environment for microbes to come up with chemical tricks to enable them to do that. So we mined all this microbiome data, and we identify individual peptides that were produced by individual microbes. And then when we synthesize them and we test them, we do MIC assays looking at antimicrobial activity against microbes that live in their surroundings. We see activity. So we think this is a form of sort of chemical warfare that microbes are using to sort of take over particular areas of and microbiome communities and sort of reconfiguring microbiome communities. And there's a lot more work to do there, but this is kind of how we're thinking about it. And some of them were effective in preclinical mouse models as well. They were safe and effective. The top candidate in this case was Prevotellin 2, which is a molecule produced by the gut microbe Prevotella copri that we all have in our intestines. So another great reminder of the amazing capabilities of microbes to come up with solutions, right? Solutions for them, but also maybe solutions for humans. You know, who knows, maybe one day we can use some of these microbially produced antibiotics to solve our own infections, right? And just very recently, we're able to complete our exploration of the tree of life by digitally mining all the archaea available in the world, and doing that digitally, of course, with AI. And this work enabled us to identify a new class of antibiotics that we're calling archaeacines. And we synthesized a lot of them using chemistry, and the hit rate was 93%. So our algorithms are getting better and better at identifying active compounds that are active actually in ground truth experiments. And I think that if we get consistently above 95%, we may not need to do as many experiments in the future. We may be able to sort of rely more heavily on these algorithms. So sort of like the last kinds of things that I wanted to tell you about is a couple of really recent projects. One of them is Apex Go. Go stands for generative optimization. So this is a generative AI algorithm that you can use to optimize your antibiotic compound. So to illustrate this practically, you can start with a template antibiotic, that is not a great antibiotic. In fact, it's a pretty weak antibiotic. And through Bayesian optimization, we can explore sequence space around this antibiotic, this template, and the algorithm generates synthetic versions of the molecule. It kind of generates new types of molecules that it predicts are going to be better than the template at killing bacteria. This is using generative Bayesian optimization. This is the first, from a computer science perspective, is the first experimental validation of this technique in any setting. And this is theoretical work, but on the experimental side, 85% hit rate. So 85% of all these newly generated synthetic molecules were active against bacteria in the lab, experimentally. And I think even more impressively, perhaps, 72% of the synthetically generated molecules by the algorithm were better at killing bacteria than the template. So again, this is just the beginning, I think, right? Here we're applying this to antibiotics, but you might imagine having an immunomodulatory molecule here and trying to optimize it. You might imagine having an anti-cancer compound and trying to optimize it, or really anything, right? You just have to change your objective function. Okay, so that's one project. And the other is that we're all familiar with multimodal algorithms. You give it a string of letters, and it creates an image or a video or things like that. But we thought about bringing this concept to the world of molecules, and more specifically the world of antibiotics. Can we use AI to design molecules that have more than one function in vivo, right? And we decided to target a very specific problem that is a huge problem, and that is the problem of intracellular infections. These are bacteria that penetrate into our human cells. They hide inside our cells in such a way that the immune system cannot kill them, and conventional antibiotics cannot really kill them either. So we thought maybe we could develop an algorithm that could design peptides that could do two things: penetrate into mammalian cells, and two, kill the bacteria once they get inside the cells. So again, like a multifunctional molecule, right? And that's what we did with AI. We generated synthetic molecules that were predicted to have both functions. And we were able to then validate this all experimentally. And the top candidates, we added a fluorophore. We followed them all inside the cell. We saw them kind of penetrating into mammalian cells and then killing the infection once they got inside. Again, this is just the beginning for this. You can imagine there are many, many disease settings where you might want to have multimodal therapeutics, right? In the case of infections, you might want to kill the bacteria, but then dampen inflammation. So maybe we can start, in fact, we have projects in the lab where we're thinking about creating antibiotics that do those two things. You might imagine having an anti-cancer agent that kills the cancer cells but doesn't kill your healthy cells, or it also reduces inflammation in the surrounding areas. And again, just an idea, right, to start thinking of multimodality in how we come up with new therapeutics. Okay, so sort of to summarize the work, like I mentioned, with traditional antibiotic discovery methods, it can take many years to come up with some preclinical candidates, but it's a very painstaking process. Today, in my lab, in a few hours, we can discover hundreds of thousands of candidates. And if we really push our resources, we can go from discovery to synthesis to testing within about 1 week. So on any given day, I literally come in the morning, have a cup of coffee, and by lunchtime, the team has already discovered a bunch of new compounds to play around with, and by dinner time, a bunch more. So it's really a dream come true for a scientist like myself, who has been dreaming about this application of AI for a long time. In terms of the impact of Apex and our other AI models, people have predicted some of this, but it's multiplied the speed of antibiotic discovery by a factor of several million. We've been able to discover millions of antibiotic candidates by sampling biology digitally. We've now mined the tree of life, including living and ancient biology. People have estimated that many, many years of research time have been saved, right? Instead of years, now in a couple hours, we can do that kind of identification. And the equivalent, people have estimated that this is equivalent to thousands of PhD students working for 6 years each. So it's really dramatically reduced the time that it takes to come up with new potential candidates. This is some of our work summarized. I didn't have time to go over all of it. And these are some of the peptides that we currently have at the preclinical stage. And we're doing some chemistry on them to further optimize them, and hopefully some of these or derivatives thereof make it into the next stages of development in the future. And with that, I'd like to thank lab members, past and present, our amazing collaborators, funders, for allowing us to do the work that we do. And I'll leave you with links to our website, email, and social media in case anybody wants to get in touch. And again, thank you for this huge honor and for listening.